So the focus of my research for almost 7 years now has been on the clinical translation of two porphyrin-lipid-based nanomedicines that in our lab we collectively call porphysomes. And in today's presentation, I wanted to share some of the interesting observations that we've made in the pharmacokinetic profiles of porphysomes related to the dose and how these have affected the design of some GLP studies, as well as our first in-patient clinical trial design. But before that, I just wanted to provide a brief introduction to pharmacokinetics and what that is. So for those of you who are hearing the word pharmacokinetics or PK for the first time, PK is simply the study of a substance's movement in the body after we've administered it. PK data is typically collected by injecting a drug intravenously into an animal, and then injecting a drug intravenously as shown here, and then collecting small blood samples at predetermined time points post-administration. And from these samples, we can reconstruct the changes in the concentration or concentration of the drug in the blood or the plasma versus time, as shown in the concentration versus time curve here. The characteristics of the curve, such as the area and the slope, are then used to calculate PK parameters, such as the area under the curve, the elimination rate, half-life. And the reason why we study PK and why it's important in drug discovery and development, especially for nanomedicines, is because of their implications on the therapeutic effect of nanomedicines, their safety and toxicity, comparisons, comparing different nanomaterial types or different batches of the same nanomaterial, as well as the design of ultimately clinical trials in patients. So as I mentioned previously, I work with a unique porphyrin-lipid-based nanoparticle called the porphysome, which is shown here in the center. Porphysomes are self-assembled from a porphyrin-lipid building block, which is shown on the left, and they form these spherical unilamellar nanoparticles about 100 nanometers in size. So both the structure and the formulation of porphysomes share strong similarities with conventional lipid-based nanoparticles like liposomes, including the presence of cholesterol and pegylated lipid, which both improve plasma circulation. In fact, these liposome-like features are there by design, and they were there in order to accelerate the clinical translation of porphysomes using roadmaps that were previously established with clinically approved liposomes. So in order to establish a similarity or bioequivalence between porphysomes and liposomes in vivo, we first needed to assess the PK of porphysomes in a manner similar of liposomes previously. So in our PK experiments, we included two unique experimental features that I would like to highlight. First, we explored the PK of porphysomes in 3 species of rodent, mice, rats, and rabbits, and appreciate how the body weight between these species is vastly different, spanning almost 3 orders of magnitude, from 25 grams up to 2.5 kilograms. The second feature of our PK experiments was that we tested a broad dose range, spanning from the subdiagnostic to the super therapeutic doses. And this was necessary for us because our porphysome nanoparticles are actually nanotheranostic agents, and they require different administered doses depending on whether or not we intended imaging or therapeutic modalities. So before jumping into the data, I just wanted to take a quick moment and thank everyone who's contributed to my work that I'm presenting today. So that includes members of the Zheng lab, my research mentors and committee members, as well as the generous support of our funding agencies and our donors. So the first piece of data that I'm presenting, we have our concentration versus time curves of porphysomes in our 3 test species, our mice, rats, and rabbits. And what you see in each graph is the plasma concentration profile of escalating doses of porphysomes in each species. Now, for the purposes of comparison, we typically normalize the graphs we see in the top to create the graphs you see along the bottom. And in these graphs along the bottom, you can see how most of these plasma profiles overlap with one another, and how certain porphysome doses actually generate profiles that are different from the rest, like here and here. In fact, this is our first evidence of dose dependency. So what we're seeing here is a phenomena. It's known as accelerated blood clearance, or ABC. And this typically affects very low doses of nanoparticles. What's quite curious is to also notice that the lowest dose that we tested in the rabbit did not produce the ABC phenomenon. From the concentration time profiles on the previous slides, we can also calculate a number of dose-specific PK parameters. So on this slide, I'm showing you the four most commonly reported parameters in nanoparticle literature: area under the curve, clearance, volume into distribution, and half-life. For simplicity, I'm also showing you only the parameters calculated in the rat, which are along the top, and in the rabbit, which are along the bottom. But note that the behaviors of the PK parameters in mice were very similar to those that we saw in the rats. For all these graphs, the vertical axis is the parameter value, and the horizontal axis is our escalating porphysome dose. Now, the exact meaning of each parameter and the values are not really what are important here, but rather I'd like you to focus on the trends within each parameter. So let's focus on AUC. Here you see in the rat at the very lowest dose, it's an outlier from the rest. And in fact, this is another example of spotting that dose dependency, which we saw previously. Moving on to the clearance and volume parameters, once more, we're easily able to identify the dose dependency in the rat data from the ABC phenomena, right? In fact, the elevated clearance at these very low doses is the reason why we call the phenomena accelerated clearance. But did you also notice that there's actually something off at the high dose in the rabbit? So here's another piece of evidence for another type of dose dependency, which we call saturable or non-linear PK. And this typically affects very high doses of nanoparticles. Non-linear PK behaviors are not ideal because their presence usually curtails our ability to relate administered nanoparticle doses to pharmacodynamic and toxicological findings. What was interesting is that we're able to detect the saturable PK parameters in the parameters, but we weren't really able to observe it in the PK profile. And finally, looking at the pharmacokinetic half-life, the dose dependency trends that we saw very obviously in the AUC, the clearance, and the volume are not really obvious here. And this seems to indicate that the porphysome's half-life might not be, or might be relatively insensitive to administered dose. So to summarize our findings, we first saw that the dose dependency in porphysome pharmacokinetics occurred at the extremes of the dose range that we tested, either very low doses, where we saw ABC phenomena, or very high doses, where we saw that nonlinear behavior. Now, these observations are likely true for most nanoparticle types and not just porphysomes. Secondly, we saw that the accelerated blood clearance, or ABC, was easily spotted in the concentration time profiles, as was the case in the rat data. But the exact dose at which ABC is triggered in each species is likely species dependent and species specific. And lastly, we found that each PK parameter exhibited a varying degree of sensitivity to dose dependencies for both at low doses and high doses. And what I mean by that is, relatively speaking, when we looked at just half-life, it was the least sensitive and least predictive of dose-dependent behaviors in porphysome PK. Whereas looking at the area under the curve, the volume, or the clearance, these were more sensitive and more predictive of these dose dependencies. So what do these findings mean? Well, you recall again that the PK concentration versus time profile, or the PK parameters, they're widely used for assessing the performance and safety of medicines. Thus, the identification of PK profiles has far-reaching implications for the selection of rodent species, right, to assess PK, as is the case in bioequivalency studies, interpretation of safety and therapeutic data, or, as was our case, the establishment of the safe starting dose for our first in-patient clinical trials. In our program, using the data that I presented to you here today, we were able to conclude that the rabbit and the rat data were our pivotal species for studying porphysome PK, safety, and therapeutic efficacy as we translate the porphysomes into first in-patient clinical trials. I'd like to thank you all for your attention, and if you have any questions, I'd be happy to answer them.
Great, thank you, Mike. And so I'm pleased to open the floor to questions from the audience. And while we give people a moment to type or put their hands up, Mike, I'd love to ask you a question. I was wondering, you probably couldn't go into this that much because of the time limit, but could you speak more about how you calculated the PK parameters?
Sure. So I did not go into it. We use one of the most simplest techniques, which is known as non-compartmental analysis. The reason why we choose this type of technique is because it's the most accessible, it's robust, it's computationally simple, and has the least number of assumptions. There are opportunities to use more advanced and sophisticated, pharmacostatistical tools. The challenge there is that you really need to understand the way you're analyzing your data and the suitability of your data for a particular tool, and also the suitability of the tool for your research question. So for our purpose, it was non-compartmental analysis. It was the simplest tool that one could choose. In the future, we do actually want to explore how the answers we found with non-compartmental analysis might change had we chosen a more sophisticated tool and perhaps an inappropriate tool, which might be the case if we didn't know what we were doing. So that's something we're exploring in the future.
Great, thank you. Hayden, are there any questions at this time?
No questions at this time.
Great, well then I can ask another one. Mike, I was wondering if you could expand on how you figured out doses between the species with different sizes and I would imagine different metabolisms.
Yeah, that's a good question too. So for most preclinical nanoparticle literature, you typically take your dose and then you normalize it to either the body weight of the animal, the surface area of the animal, or if you want to be a bit more exotic, you can actually look at specific organ weights or blood flows or even cellular populations. So that's the most conventional way. But I think in the field where we're actually seeing a lot of the work go, particularly the PK, nanomaterial PK, which is also a direction we're trying to move towards, is actually relating function between different species as opposed to just normalized dose. So what that means is relying on the imaging dose, the optimized imaging dose in each species, or the maximum tolerated dose if you're looking at toxicity between species, and using that as your functional equivalence in a way of comparing doses. It is a challenging problem, but I think the simplest, as we did here, was just the number of nanoparticles per kilogram.
Great, thank you.